all yours.
Okay. It's really a privilege for me tonight to introduce Dr. Murray, a dear friend of mine, and I cherish each one of his presentations. But I want to give a little background on Dr. Murray. He was born in Scotland. He received his MD degree from the University of Glasgow, Scotland. He interned at Victoria Hospital in London, Ontario, and then completed a further year of postgraduate training at the University of Michigan, USA, where he was awarded an MS in a master's degree in physiology. This was followed by 3 more years of graduate research in biochemistry at the University of Toronto in Canada, culminating in him being awarded a PhD degree, and I assume that was in biochemistry. Dr. Murray had a distinguished teaching and research career at the university, during which he received faculty and alumni awards for teaching, trained more than a dozen graduate students, published over 50 scientific peer-reviewed papers, authored multiple textbook articles, and has been the senior editor of the last 7 editions of Harper's Biochemistry. And I just want to mention, I've been told that Harper's Biochemistry is taught in 82 of the 120 medical schools in the United States. So that'll give you some idea of what a privilege it is for us to hear this presentation tonight, and I turn it over to Dr. Murray.
Thank you. Are we all set, David?
Yes, sir, we are. All you need to do is make it a slideshow, and right there, yeah, just to the left. There you go. You got it.
This one?
Yes, sir.
Yes.
Okay. So, yeah, thank you, David, and thank you, Dr. Marvin, for your very gracious introduction. I'd like to thank David and Sherry for inviting me to speak again. Good evening to everyone who's listening, and many thanks for joining us. I'm up outside Toronto. It's a cool evening up here, and we had some light snow today. I don't know what it's like, obviously, where you listeners are. I guess it's quite a bit warmer than here. So tonight I'm going to be talking about aspects of basic genetics. There'll be quite a bit of chemistry I'll be showing, but you don't have to remember the details. But I think without showing the chemistry, you would not really get much of an idea about genetic material. So I apologize for that, but don't obsess on the details. The idea is to get the general picture. So this slide gives a very, very brief history of genetics. Apologies to many people who made important contributions. I've only shown a few major discoveries or observations, and I guess genetics really started with the work of Gregor Mendel, who was a monk in Brno in what is now Czechoslovakia, and he did classic experiments involving breeding peas that clearly showed the existence of hereditary factors. And there were many other important discoveries subsequent to that, but the next key observation was made by researchers in the early 1940s at the Rockefeller University in New York, Dr. Avery and colleagues, and they showed that the genetic material in the organism they were studying, which was a bacteriophage, a virus that infects bacteria, was DNA. And then pivotal observations were made in 1953 by Watson and Crick. Watson, an American, Crick, an Englishman, working at Cambridge University in England, when they showed the double helical nature of DNA, and that helped explain important aspects of heredity. So that was certainly one of the most fundamental series of observations in the history of genetics. Then about 1990, the International Human Genome Project involving laboratories in the U.S., the U.K., Japan, Canada, various countries in Europe, etc., was started, and it concluded more or less in 2003. I'll be talking about it tonight in a little bit of detail. And the work of this very important international project showed that human genetic material, that is the human genome, was comprised of some 3.3 billion base pairs. I'll explain subsequently what a base pair is. And much of the sequence of these 3.3 billion base pairs, that is the way they're linked together, was determined. So apologies to thousands of other important contributions to genetics. But these are certainly, I think, the two key observations, Watson and Crick's work and the work of the Human Genome Project. I don't think the slide is advancing here.
Have to right-click on that screen and select Next.
Okay, thank you. Do I right-click every slide?
No, sometimes you just need that to do that just to get it to free it up.
Okay, so here we, thank you, David. Here we see a relatively simple diagram of a cell, a human cell. This is the surface or plasma membrane. This is the nucleus where the genetic material is. In the case of humans and most animals, it's DNA. And as we'll find out, DNA can be copied to RNA, and the information in DNA is retained in the RNA copies, and the various copies make proteins in the human cell. Something like 26,000 proteins are coded for by our genetic material. And the RNA will leave the nucleus, this is the nucleus, and travel either to little ribosomes, which are free in the cytoplasm of the cell, or to ribosomes that are bound to the membranes of the endoplasmic reticulum. And on these ribosomes, the free and what we call the bound ribosomes, proteins are made, and then they're distributed to different parts of the cell. Some travel to the surface, some travel to this organelle, the lysosome, some travel to all different parts of the cell, okay? So the information for the coding of proteins is in the DNA. It is transferred, if you like, transcribed to RNA, species of RNA, and then the RNA is used for protein synthesis on these little particles called ribosomes. These are free ribosomes, and these ones here are bound to the membrane, so they're called bound ribosomes. Yeah, I'm having trouble switching the slides.
I have to do that again. Sorry about that.
Yeah, I did it. Okay. So the next question then is, what is a gene? And you might be puzzled by the fact that there is no entirely satisfactory definition of what a gene is. The simplest explanation is that a gene is a fundamental unit of heredity determining one aspect of heredity. In most cases, a gene is in fact the sequence of the DNA in the chromosomes, that is in the nucleus where the chromosomes are, that encodes or directs the sequence. By sequence, I mean the sequence of amino acids in a protein. And as you may know, our proteins are made up of amino acids, and one protein differs from another in its sequence, okay? Some, like insulin, contains about 51 amino acids. There's another protein in muscle that contains some 28,000 amino acids. The information for the sequence of amino acids is coded in the DNA, transferred to RNA, and in turn that makes the protein. So the central dogma, this was coined by quick of molecular biology is DNA transferring its information to messenger RNA, which is one type of RNA, and in turn that codes for proteins. And the mRNA is a copy of part of the gene, okay? And it encodes or directs the sequence of proteins. So it retains the genetic information that is in the DNA, and eventually that governs the synthesis of the whole array of proteins that are made in human cells, at least probably 25,000 and perhaps quite a bit more. The mRNA is a copy of part of the gene, okay? So RNA is ribonucleic acid, and the difference from DNA is it contains a sugar called ribose that I'll show you subsequently, not deoxyribose. DNA contains deoxyribose, RNA contains ribose. They're both simple 5-carbon sugars. And then its bases, these are key molecules in both RNA and DNA, are adenine, guanine, cytosine, and DNA contains thymine. These are the 4 bases in DNA, adenine, guanine, cytosine, and thymine, whereas in RNA, uracil, this base replaces thymine. So the difference between RNA and DNA is one in the sugar. Okay, ribose contains, RNA contains ribose. On the other hand, the difference in bases that RNA contains uracil, not thymine. And I'll show you structures of these compounds in a minute, which should hopefully help clarify what I'm saying. Okay? Now, as mentioned above, usually the product of a gene is a protein, or it may be a chain of a protein, because some proteins are made up of several chains, and one gene may encode the synthesis of one chain and another gene of other chains in the protein. So each chain may be encoded by a separate gene. Okay? It seems like I have to right-click all the time, but that's okay. So carrying this further, what is a gene? The product of a gene may be an RNA molecule. In some cases, the gene does not code for a protein. It codes for RNA molecules, and there's tremendous interest in these molecules because they've only been discovered relatively recently, in say the past 10 years or so, and these help to regulate the functions of genes. They're regulatory RNA molecules, and they are different from messenger RNA that I already talked about. This is this class of molecule in which there's tremendous interest in at the present time. Any of you who are interested in molecular biology will know that this is a major focus of research at the present time. Now, another aspect is that the mitochondria, which are little structures inside the cell that were shown back in the slide of the structure of the cell, they have their own genes which are not located in the chromosomes, and they form circular molecules inside the mitochondria. So there's a nuclear genome where the mass of the genes are, and there's a mitochondrial genome which is much smaller but is nevertheless important. Then another factor that complicates the discussion of what a gene is, that viruses have genes, okay, which are not organized as chromosomes, and in some viruses, RNA is the genetic material. For example, in the influenza viruses that we're all very aware of, and HIV-1 and 2, which most people think cause, if not play a significant contribution to AIDS, their genetic material is RNA. So that's why I hedged a little in the definition of what a gene is. Okay? So in the next slide then, in this slide we trying to move this out the way. The next question is, of what are genes made? Okay? And I've adopted this rather ugly English construction because we don't like to finish a statement with a preposition, as Mr. Churchill said at one time. Okay, so anyway, human genes and those of most species are made up of DNA. So there's three important types of constituents in DNA. The sugar, deoxyribose, four bases, adenine, thymine, guanine, and cytosine. They all differ somewhat from each other. And then the third important constituent is phosphate. So these are the basic constituents of DNA. Okay, so deoxyribose, one of the four bases, either deoxyribose will link up with adenine or thymine or guanine or cytosine and one phosphate to form a compound called a deoxynucleotide. A nucleotide is a combination of a sugar, a base, and a phosphate. And if the sugar is deoxyribose, we call this a deoxynucleotide. And in fact, DNA is made up of millions and millions of deoxynucleotides. So the basic building block is the deoxynucleotide. So there's a base, these four bases, A, T, G, or C, the sugar, deoxyribose, and the phosphate. And that forms a deoxynucleotide, which is the basic building block of DNA. A base, deoxyribose, and phosphate. And thousands of these nucleotides link together in a specific sequence to form one gene. And each gene has a different sequence. So there's something like 26,000 genes in humans, and in the Human Genome Project, the sequence of almost all of these genes was determined the exact way the nucleotides are linked together. And that was a tremendous feat to do that. Millions of nucleotides linked together to form one large molecule of DNA which contains many different genes. And one large molecule of DNA bonds or pairs with another large molecule of DNA to form the double helical structure described in 1953 by Watson and Crick. Okay, so the next question we address is, what is a genome? Okay, well, the genome is all the DNA, or if it's a virus containing RNA, all the RNA in an organism, including its genes. But it's interesting, in fact, that only about 3% of the DNA in our cells are genes. The other 97% have other functions. They do not code directly for proteins. Genes usually encode proteins which do the work of the body. The DNA, if you like, is the master molecule that encodes the proteins. The working molecules are the proteins, which are made up of chains of amino acids. They do the work of the body. So the genes, if you like, are the templates from which proteins are constructed, the mold or templates from which proteins are constructed. And as I said, DNA is made up of 4 different deoxynucleotides, each containing one of the 4 different bases, A, adenine, thymine, cytosine, or guanine. So we'll just call them A, T, C, or G from now on. And it's the order of these 4 bases in a gene that dictate whether an organism is a human or some other species. So one can consider that, depending on your philosophy or religion, as the way God made our DNA. But this is why it's important to determine the sequences of the bases in DNA because it is the sequences that determine what type of organism we're dealing with, whether it be human or some other species. The simple representation of the double helix as first elaborated by Watson and Crick. They had, I won't go into the debate of the role of other people in that work, Rosamund Franklin and other people. She certainly had an important role in it, but I'm not going to discuss that tonight. But here we see one molecule of DNA, and here we see the other molecule of DNA. And what we see is these are the bases A and T, T and A, okay, C and G, G and C. S is the sugar, deoxyribose. P is the phosphate. So there we have the phosphate sugar base. That's a nucleotide, okay. Here we have another nucleotide, phosphate sugar base. And you'll see a key point that Watson and Crick really determined, although there was previous experimental work pointing to this, is that adenine always bonds with thymine, A with T, or T with A, that's the same, both adenine and thymine. And cytosine, one of the four bases, always bonds with guanine, guanine with cytosine. And once they discovered this, this was a key observation in leading them to formulating the double helix. So this helix is a very long molecule, and thousands and thousands or even millions of nucleotides in one chain. And that is a nucleotide, as I said, base, sugar, phosphate. So we're going all the way down here. There would be thousands, many, many thousands of bases making a molecule. And the precise dimensions of the helix were determined by them too, that it was about 20 angstroms, that's a unit of size, in width, and one turn of the helix took about 34 angstroms. But a key thing is that adenine always pairs with thymine, and cytosine always pairs with guanine. This is called base pairing. So a base pair, these two molecules here form a base pair, adenine and thymine. This is another base pair. So when I say that there's 3.2 billion base pairs in DNA, that means there's 6.4 billion bases, but we talk about base pairs, okay? So in human DNA, there's about 3.2 billion base pairs, with this being a base pair, this being a base pair, and this base pairing rules always hold. T with A with T, C with G, G with C, but never G with A and never G with T, and vice versa. Okay, so this is really the fundamental double helix that was postulated by Watson and Crick. They built many models and came up with this structure. And I think this had a revolutionary effect on the science of genetics. There's no doubt about that. Okay, so here we see then, just to give you an idea of the structure in a little more detail, forget the details, but this is the structure of guanine, the base guanine. There's the sugar, deoxyribose. There's the phosphate. Here we see cytosine. It's a different structure, okay? We're going along one strand of DNA, okay? Here we see thymine, which has got a different structure from cytosine. And here we see adenine, which is similar to guanine, but again has a different structure. So this is a nucleotide, a base, a sugar, and a phosphate. This is another nucleotide, base, sugar, phosphate. These are 4 nucleotides, and DNA is made up of millions of nucleotides joined together in a specific sequence. The sequence is not random, it is quite specific. And as I'll tell you later, the sequence of 3 bases ultimately encodes for 1 amino acid. Okay, we'll come to that later. Yeah, it's a pity that I can't advance. Don't seem to be advancing properly here. Can you help me there, David? Or
David?
Yes, I'm here. Can you try left-clicking on the screen itself. Let's see if that works.
Excellent. Does that sometimes do the trick?
Yeah, it's you just have to keep trying the options.
Okay.
Use the
Yeah,
arrows.
stick around.
Okay, I will. I will.
Okay. So here, just to get this principle clear of what a nucleotide is, here are two different nucleotides. They're both made of phosphate, sugar, and base. This is a nucleotide found in RNA, in which we have adenine, okay, and then it's called AMP, adenosine monophosphate. Adenosine means it's adenine linked to the sugar ribose, and then the phosphate. So this is a nucleotide here. And in ribose, the only difference between ribose and deoxyribose is here. You see that in deoxyribose, as shown here, there is no oxygen in this position, hence the name deoxyribose. They're both ribose, if you like, except in this case, the oxygen has been taken out. So this is a nucleotide found in DNA. It's a deoxynucleotide, and this is a nucleotide found in RNA. It's AMP. This is deoxy-AMP because this sugar is deoxyribose. Okay, so here we see then, I'm just trying to pin this down because the base pairing is critical to understanding all of molecular biology, really, DNA and RNA. But here we see thymine, this is thymine, pairing with adenine. I told you adenine and thymine always pair. And together, these are about 20 angstroms wide, okay? C and G always pair together, and if T did pair with C or A with G, that would not fit into the 20 angstrom wide structure. So this is part of the reason these together form a structure of about 20 angstroms wide as do these, okay? So the T always pairs with A, and C always pairs with G, and C does not pair with T or A or vice versa. Okay, now the beauty of the model that Watson and Crick proposed was that it helped explain the replication of DNA during cell division. These are the two strands of the double helix. When during cell division, when a cell is multiplying, the strands separate, and in the one strand, wherever there's a T, an enzyme will add an A, in this case an A here, or a G. So on the one strand, the perfect model, the other strand is laid down, and similarly in this strand, because of the base pairing, wherever there's a T, an A here, a T is laid down, and an enzyme builds up a new strand of DNA. So after cell division is completed, you have doubled the DNA, and that will pass into the daughter cells. So a beauty of their molecule was that it helped to explain the replication of DNA at cell division, and this depended on this base pairing. The new strands of DNA were constructed from the old strands by an enzyme that built up the new strands, but following the base pairing. Wherever there was an A in the old strand, a T would be built up in the new one. Wherever there was a G in the old strand, a C would be built up in the new one. So this demonstrated the way that the fidelity, if you like, of the genetic material could be replicated at cell division. And that was a key discovery. Prior to this, it was not known how the genetic material was faithfully replicated in cell division. Okay. Now, to just give you a little bit further detail, if we take the two strands of DNA, this is one strand here with the bases shown, T, G, etc. This is another strand, okay. These are the direction we talked about the 5' end and the 3' end okay, and the other strand runs the other way. This strand is running that way. The second strand runs the opposite direction, but this strand here is used to make an RNA copy, okay? Wherever there's a C, a G is made. Wherever there's an A, because RNA does not contain T, it contains U. A U is laid down. C goes to G, okay? T leads to A, etc. So this RNA copy is made from this strand, but in fact it retains the information in the so-called coding strand, although you must remember that we changed T to U, okay? So the legend to this slide explains that. This is the template strand from which the RNA is made, but it preserves the information, the genetic information in the coding strand, with the exception that U replaces T, okay? So this is the way the information from DNA gets into RNA, and ultimately it's going to make protein.
Now, excuse me, but is the 5 and 3 strand, or you're going to explain it?
Well, I won't go into that in a lot of detail, but the basic concept is that one DNA strand goes from the 5' end. There's a difference at this end to the 3' end, and then the other one runs in the opposite direction.
I see. Yeah. Okay.
But the base pairing, is preserved, A, T, G, C. But the two strands are running in opposite directions. So.
Okay.
A messenger RNA, good question though, the messenger RNA is made from the one strand, okay, then it travels out of the nucleus after some changes that I'll talk about briefly, then it gets to the ribosome. These are the ribosomes, these little structures. They're made up of protein and other types of RNA molecules. Some are free, sitting in the cytoplasm of the cell, others are bound to the endoplasmic reticulum. And the protein starts to be made, and here we see it elongating, getting longer, and then it's finally released from the ribosome, and then the protein can travel to different parts of the cell. It can go to the surface membrane, it can go to the lysosome, it can go to the cytosol. It has different fates inside the cell.
Okay.
Okay, so here's the mRNA here, the messenger RNA, the copy. Okay, the sequence in RNA, this was a very important discovery by an American biochemist who just died, Marshall Nirenberg, working at the NIH in Bethesda, Maryland. He and his collaborators were able to show that the code for an amino acid was a triplet code, okay? And they found, for example, that a sequence of U in the RNA led to encoded the amino acid phenylalanine. That's one of the 20 amino acids in proteins. Glycine, valine, methionine, there's a whole bunch, 20 amino acids. And the code was in the RNA, and this molecule here is another type of RNA molecule called transfer RNA, and at the end of it, the amino acid was attached, and gradually in different parts of the RNA, there was a code for different amino acids, and the proteins could then be built up. Here with phenylalanine, the next amino acid might be glycine, coded by a slightly different codon made up of Phe bases, etc., so that in the case of insulin, there'd be codons for, well, actually over 51 amino acids without going into the details, etc. But so this is the mRNA. It interacts with another type of RNA molecule called the transfer RNA, and at the end of it, the amino acid is present, coded, in this case, phenylalanine, by this triplet codon. All of the amino acids are coded by triplets, three bases, but the code for each one is different. For another amino acid, it might be AUG. For another amino acid, it might be GUU, etc. So in total, there's about 60 different codons coding for the 20 different amino acids. Some amino acids have more than one codon without going into details.
What is the difference, Dr. Murray, from the PU and the PY?
Where are we not? Yeah, I should have explained that probably. Purine is the simple base.
Right.
Both thymine and, well, let me put it, both adenine and guanine are purines, whereas thymine and cytosine and uracil are pyrimidines. They're all bases, but there's two. You might have noticed if you went back to the structure of adenine and guanine that they had 2 rings, whereas the pyrimidines only have 1 ring. You'd have to go back to an earlier slide. But PU is purine, PY, but basically that's a detail, but they're just types of bases. Okay?
Okay.
Yep. Thank you. Okay, now I'm not going to go into this in detail, but one of the problems with DNA, it can be damaged in many ways. It can be damaged by chemicals interacting with it, by radiation can break it, etc. There's a heavy dose of radiation, and these damages can lead to single base alterations. We don't worry about the details of the chemistry, but these are some of the changes that can happen without going into details. Okay, single bases can be changed. You can have 2 bases being changed. For example, UV light can exposure of DNA to ultraviolet light can lead to 2 bases being changed. Certain certain anti-cancer drugs are alkylating agents, so they can, in some cases, a high dosage lead again to damage. Chain breaks can be, the DNA chains can be broken by ionizing radiation, okay, by free radicals, etc. Okay, and in severe damage, you can also get cross-linking of the DNAs, not the normal ATGC, but actual chemical bonds can form between the 2 chains, and that is quite damaging because it will prevent the strands separating. So don't worry about the different types, but the concept is that DNA can be damaged in a variety of ways by chemicals, by UV light, by radiation, etc. Okay? And in fact, DNA can be repaired. There are enzymes in our cells that will repair DNA, although in some cases if the damage is heavy, the damage will not be repaired. Let's imagine that a chemical lands on this part of a DNA molecule here. There's one strand, okay, there. There's another strand here. Okay, if there's damage here, then an enzyme which is present in the nucleus can recognize this site, and then it can chop up this area that includes the damaged area. Okay, it's eliminated the damaged area. Then another enzyme comes in that's called a polymerase and rebuilds the damaged area. And here, if all goes well, we have restored the structure of DNA. So this is called DNA repair, and it can be, if the damage is slight, successful, but if the damage is heavy, the DNA may not be repaired, and this can lead to permanent mutations. Okay, so the enzyme that chops out the damaged area is called a nuclease, an endonuclease, because it's working inside the chain of DNA, not at the end of the DNA. Nuclease because it's working on nucleic acid. DNA is a nucleic acid. Okay, then the enzyme that builds up is called a polymerase, etc. And then this piece is then tied into this piece by another enzyme called a ligase that binds this end here to that end, this end to that end. So you have a variety of enzymes involved in DNA repair, and this is a very important process because if the enzymes of DNA repair do not work properly, this can lead to permanent mutations, which in some cases, for example, can result in cancer. Or if you're getting excess exposure of UV light by tanning procedures, this can lead to mutations which may, in some cases, result in skin tumors such as melanomas. Okay? So DNA can be repaired, but it's not always perfect depending on the extent of the damage. Okay? Now here we see then a picture of the production of the messenger RNA. This is a chromosome which can contain a large number of genes, okay? It's made up of many base pairs, 1.5 times 10 to the 8 base pairs in this particular case. Here we see this little segment of the chromosome here magnified, and we see there's about 20 genes in this particular area. Okay, these are indicated by the black lines. This is one gene, another, etc. So there's about 20 genes in this cluster. Okay, then if we pick out this particular gene, okay, we see that this is the structure of this one particular gene. So we're going from the chromosome to a cluster to a single gene. Then this gene is copied in the process that I already told you about, making RNA, and the copy is called the primary transcript, the first product. And when you copy DNA to RNA, that is called transcription. Okay, DNA being copied to RNA is transcription. So we have this RNA molecule, but amazingly, the final RNA that codes for the protein, these areas here are cut out by enzymes, these gray areas, so that the final mRNA that codes for the protein contains this segment, this segment, this segment, this segment, this segment, linked together to form this particular messenger RNA. So you can see this is a pretty complex process, okay, in which the gene is copied, but then parts of it are chopped out, okay, and you have the final molecule that will code for the protein, okay. And this, what I'm presenting here, in a minute or two, it took people 20 years or more, many investigators in different parts of the world, some of whom received Nobel Prizes for determining this particular pathway of how the messenger RNA is made. Okay, so I think now we should have some idea of the structure of DNA and of RNA, okay, without worrying or obsessing about the chemical details. But here we know that there's 23 pairs of human chromosomes, okay, and these can be seen by microscopy. They can be brought out, made accentuated by procedures so that they're easily recognized. And chromosome 1, the two chromosomes are the largest, okay. This is chromosome 2, 3. They're sorted in terms of length. And down here we have the sex chromosomes, the X, which is much bigger than the Y, okay. So we call these 1 to 22 are called autosomal, and there's one pair, X and Y, that are the sex chromosomes. So in a female, you would have two X's. In the male, you have X and Y, and the Y is different from the X. It has a lot less genetic material than does the X, okay?
Does that mean that females are more complicated? It takes a bigger chromosome to make a female?
Well, I'll leave you to make up your mind whether females are more complicated. I don't want to get engulfed in that particular discussion, but certainly the genetic material in the X chromosome is a lot more than the Y chromosome. Maybe that's why men are so simple and etc. But I don't want to get into that.
Okay.
Okay. Good question, though. So now we come to the Human Genome Project. When it started, Watson and Crick, of course, had done their work. The general structure of a gene was known. The DNA to RNA pathway had been worked out. The general principles of how proteins are made in cells had been worked out. But the overall aim of the Human Genome Project was much grander, if you like, Napoleonic in scale. It was to identify all the genes in human DNA and, in fact, to determine the precise structure of all human DNA. And we now know that only about 2% is genes. The rest is other functions, regulation, various other functions. So this was one, to identify all the genes. It turned out there's about 26,000. Almost all the sequence of the DNA, 3.2 or billion base pairs, okay, was more or less sequenced, okay. The information was stored in computers, databases, so that different laboratories and across the world could access it. Then various technologies stemmed from the Human Genome Project, okay. So one purpose was to transfer related technologies to the private sector, and in fact the Human Genome Project, which involved determining the structure of the genome, that's the science of genomics, it also led to other related sciences like proteomics, which is the study of all the proteins, for example, in humans, or all the proteins of cattle, or all the proteins of any organism. That's called the proteome, the complete complement of proteins in an organism. There were also important ethical, legal, and social issues that had to be considered, okay? And also, at the same time, the genomes of several non-human organisms that had played a central role in genetics, for example, the bacterium E. coli, the fruit fly, Drosophila melanogaster, and the laboratory mouse, because the laboratory mouse is particularly suitable for manipulation of genetic material. These also were aims of the Human Genome Project, to sequence the genomes of these particular species. And since the completion of the human genome species, many various sub-branches related to genomics, the study of genomes, have emerged. For example, glycomics, the study of sugars, really was spurred on by the Human Genome Project, as with the study of many other sub-areas of science have been greatly stimulated by this project. Okay, so what were the major findings of the Human Genome Project? Okay, the human genome contains approximately 3.2 million base pairs, so 6.4 bases, because a base pair is ATGC, so actually there's 6.4 billion bases, but we talk about base pairs, okay? I wanted to clarify that for you. The average gene, if you can think of an average gene, consists of about 3,000 base pairs, okay? But sizes vary widely. For example, this one has a size of 2.4 million base pairs, a really huge gene, whereas others might be somewhat smaller than this. But this is the average size. And the total number of genes in humans is approximately 25,000, which surprisingly is not that much more than the fruit fly, yeast, or roundworm, which are somewhere in the 16 to 20,000 area. This was a major surprise because most of us think of humans being much more complex, obviously, than these relatively simpler organisms, okay? Now the sequence of our genome is identical in much of the 99%, but even 0.1% leaves a lot of area for variation, because 0.1% of 3.2 billion is still a very large number. So there are many little differences, variations among us, okay? But as I say here, 0.1% of 3.2 billion is quite a large number, permitting considerable variations among individuals, and these are being looked at in great detail by many labs across the world to see if these variations correlate with diseases, for example. Now, the functions of many genes are yet to be discovered, okay? We know the sequence of them, but what proteins, etc., they code for has not been completely determined, okay? Now, other facts about the human genome: there are repeat sequences, short and long sequences, that do not code for proteins, and they make up about 50% of the genome. I told you that only 2% directly codes for proteins. And these repeat sequences seem to reshape the genome over time by allowing it to rearrange, and new genes can be formed. The genes appear to be randomly situated along the genome with vast areas of non-coding DNA, that's at 98% material, occurring in between genes. Now, non-coding areas have been referred to as junk DNA, but this is in a sense a misnomer. It's not a correct term, as these areas have functions that have not really been elucidated as yet. For example, regulation of genetic material by these areas, or the packaging of DNA into a chromosome, because that involves packing a huge molecule into a relatively small structure. Now, chromosome 1 is the largest human chromosome. It has the most genes, and this has been exactly determined, 3,168, and the Y chromosome has the least. I've forgotten the precise number for the X chromosome. And many genes have been identified as being involved in various genetic diseases. Okay? Now, an interesting area is comparative genomics. Genomics means the study of genomes, and many laboratories are studying and comparing, looking at various plant genomes, for example, rice or any plants of great commercial and nutritional interest are being examined. The genomes of various species, dog, mouse, rat, etc. So this is part of comparative biology now, okay? Even though we have not that many more genes than other species, a phenomenon called alternative splicing allows human genes to encode for 3 or more different proteins. If you take, if this is a human gene here, okay, and you make an RNA copy of it, as I showed before, if this is the RNA copy, the way you splice or split that RNA can lead in some cases to 3 proteins being made by one RNA. Okay, so this is called alternative splicing. And this occurs much more in humans and, than in simple organisms. So we can have many more proteins than simpler organisms, helping to explain our apparent complexity. So even though we have 25,000 genes, most of which, but not all, code for proteins, some code for these regulatory RNA molecules, let's say it's 25,000, if we have a lot of these are coding for as many as 3 proteins, there may be 75,000 proteins in humans. No one yet knows the precise number of proteins in humans, and that's a very active area. That's called proteomics, studying the proteome, that is the entire protein composition of humans and other species. We have most of the same families of proteins as the fly or worm, but the number of gene family members has expanded in humans, and especially in proteins involved in development, that is embryonic development, etc., and also in immunity. We are a lot more complex than these simpler species, the fruit fly or the roundworm. Okay, I won't give the precise names of these species here right now. We have many more repeat sequences, I mentioned that before, than lower species, and people are looking at the significance of this. Okay, And another area, interesting area, is examining the genes in rather closely related species, say chimpanzees, that would help to explain the acquisition of higher intellectual functions. Are there genes and specialized areas of genes that have permitted, let me put it another way, that are involved in speech that, for example, other species cannot speak? Okay, so this is a very interesting area that quite a number of labs are looking at the present time. Now, I mentioned the variations, okay, 0.1% differs, but this means there are millions of locations on our chromosomes where single-base DNA differences have been identified, and many research projects are analyzing whether these locations are associated with common diseases, for example, heart disease, diabetes mellitus, cancer, etc. This is a very, very active area of research. For example, in the New England Journal of Medicine, which is one of the premier medical journals, almost every week there's a study now about associating variations with various diseases. Okay, now whether that's the answer, these variations are going to show up as the cause of heart disease, diabetes, cancer, is still not entirely clear. Okay, there are many genes involved in heart disease. There's probably quite a number of genes involved in diabetes, and we know that there are many mutations of genes in cancer cells, so that these are very active areas of research. Okay, so some of the major issues still to be settled are the following. We want to know the exact number of genes and also the functions of these genes. Ultimately, people would like to know the function of every human gene, what protein it's making, etc. People want to know how genes are regulated, for example, how they're turned on or off. Dr. Marvin will talk about that in nutrigenomics, how nutrients influence the activities of genes. This is a very important area in which there's a lot not known, because certain genes are turned on in the embryo and then the fetus, and then they're turned off, so that as we age from embryo to fetus, etc., to adult, various genes are being turned on or off, and people want to know exactly what regulates genes. We want to know the functions of the so-called junk DNA, which is, I mentioned, a misnomer. We want to know the size and nature of the proteome, that is, the structures and functions of all human proteins, but also in other species as well. Identification of genes involved in all diseases, particularly the important chronic diseases are now, which are placing a huge burden on health systems all over the world. There are now techniques in which on simple plastic plates, the activities of 2,000 or 3,000 or 4,000 or more genes can be looked at with techniques I won't go into tonight, gene scans, to look at changes in development, in aging, cancer, changes caused by nutrition, various diets, etc., can be examined using plates that are coated with different genes, and this can help determine whether they're active or inactive, and also comparative studies of other species, animals, plants, bacteria. Let's say, for example, the influenza virus, its genome has already been determined, and as I mentioned earlier, the influenza viruses are RNA genomes. Okay? So some practical benefits of the Human Genome Project are, in humans, it's of help in diagnosis. There are now methods of looking for abnormalities in various families. If it's suspected there's a genetic disease, you can have probes that will probe whether a gene is altered or mutated. The whole area of treatment, okay, using genes, gene therapy, this is called, is being actively investigated, and There's a few breakthroughs already being made, but people are hoping that knowing the genome will open up the treatment of the thousands of different genetic diseases that exist, okay? Now in non-humans, the information from the Human Genome Project is being applied to a variety of challenges, knowledge of bacteria and viral genes for infections, as I mentioned, the influenza viruses, E. coli, various bacteria that are highly pathogenic. We want the people, by studying their genes, will be able to come up with information as to how they're infectious, etc. Applications to agriculture, rice, and other crops, their genomes are being determined or already have been determined. Energy production, if you live in Iowa or other parts of the states, corn and ethanol production, etc., then there's also hope that specific microorganisms can be utilized to clean up hydrocarbon and other chemical spills. And a lot of the impetus for development of these fields has come from the Human Genome Project. Okay, so I'm just going to mention briefly the nature, the major causes of genetic disorders. And in fact, there's 4 types of causes. One is a single gene, okay, where there's a mutation, one mutation in a gene that alters the gene structure, that alters the messenger RNA made from that gene, and that alters the protein, and the protein that's made no longer does its function, and that's what causes the genetic disease. Then most of the chronic diseases that I mentioned, heart disease, rheumatoid arthritis, etc., diabetes, they appear to be multifactorial, involving many different genes, polygenic, many genes. They're complex, whereas these are relatively simple. Okay, then there are chromosomal translocations, abnormalities of chromosomes. For example, many of us know about Down syndrome, where there's an extra chromosome 21, okay? And then there are also diseases due to mutations in mitochondrial DNA. As I mentioned, the mitochondria has its own genome, a very small genome, but it codes for a minimum of 12 proteins or more, and a variety of genetic diseases due to mutations in mitochondria have been reported. Okay? So what is a mutation? It's any permanent heritable change in the sequence of DNA, although in a virus it could be RNA, as I mentioned before. The causes can be exogenous, such as radiation, chemical, or spontaneous, not known to be caused by an exogenous agent. And there are many types of mutations. For example, simple base pair substitution, changing one base, adenine for guanine, etc. Okay? In some cases, a mutation may not be harmful. It may be silent. It may have no effect, depending on the amino acid and the protein that is changed, or where the mutation occurs, if it occurs in a non-coding area of DNA. Or in some mutations can change the one amino acid by changing the code. You can change one amino acid for another. You're changing the triplet code for an amino acid to another one by changing one base. Okay. You also have nonsense mutations which stop the protein being made. They interfere with the growth of the protein. No protein is made, and that can have very serious effects if the protein is playing a key role in the cell. So you can also have deletions where part of a gene is deleted out. Okay, it may just be 1 or 2 bases, or it may be a much bigger section of the gene. You can have insertions of genetic material, new material being added into a gene, and in some cases you can have duplications of a gene. Okay, then there's another type of mutation in which a small area is repeated over and over again, and this is called extended repeats. It might be one base is repeated over and over again, and that's giving extra genetic material, but that interferes with the production of proteins because you've interfered with the genetic code. So some do not cause disease, depending on where they're located or the function of the protein, whereas others, of course, do. Okay? So single gene disorders are probably the simplest to understand, the most studied, and these are caused by changes in DNA sequences of a single gene. And as you probably know, I'm not going to go into this in great detail, they're classified as autosomal dominant. Okay, autosome was the non-sex chromosomes. If it's dominant, if one of there are two copies of a gene, dominant means that if one copy of the gene is mutated, you can have disease, okay? And the statistics of this happening are 1 and 2. Recessive is both copies of the gene have to be mutated, and there's a 1 in 4 chance of that happening. And some are linked to the X chromosome, for example, the bleeding disorder, hemophilia. Okay, so autosome is an ordinary non-sex chromosome. I mentioned that. X and Y are the female and male chromosomes. There's 23 pairs of chromosomes. 22 are autosomal. Females XX, males XY. The normal gene makes its messenger RNA. That makes the protein. It functions normally. We have wellness. If we have a mutation in the gene, we get an abnormal RNA that gives an abnormal protein, does not perform its normal function, we can have a genetic disease. Over 6,000 single gene disorders are already known. About 1 in 200 births is a single gene disorder. Monogenic means single gene, and this is classical genetics going back to Mendel, whom I mentioned at the beginning. And some examples of these diseases are cystic fibrosis, sickle cell anemia. One change of a base can make a different hemoglobin, and that predisposes the patient to, causes the disease sickle cell anemia, which can be mild or it can be very severe. Marfan syndrome is a disease of connective tissue protein. Okay, some people believe that President Lincoln had this particular mutation because he was tall and he had a particular structure, anatomical structure that is often seen in patients with Marfan syndrome. Huntington's disease, Woody Guthrie, the folk singer, had this. There's now a genetic test for this that you can be tested for if you believe it runs in the family now. There's also ethical considerations there. Do you really want to know at the age of 20 that when you reach 50 you're going to come down with Huntington's disease? Some people want to know that, others might not want to know it. And a very important, one of the commonest of all genetic diseases in North America, in the US and Canada, which came out of Europe, Northern Europe, is this disease, hereditary hemochromatosis, which is accumulation of iron in the body, damaging various organs of the body, okay? And a couple of genes have been implicated in this particular disease. I have a friend here in Toronto. He's of Scottish origin, and he has a very severe case of hereditary hemochromatosis. But surprisingly, this is, I think, even more common in North America than is cystic fibrosis. Dr. Marvin, have you seen cases with hemochromatosis, Dr. Marvin?
Yes, I have, and I think just what you said, it's more common.
More common.
Than cystic fibrosis.
Yeah, and there are tests for. Sorry.
I had a question.
Yes.
You were muted here a while, or I was muted a while ago, but now I'm open. I wanted to ask you a question. I understood when you talk about the mitochondrial genome, that we all get our mitochondrial genes, or the genomes, all come from our mothers.
That's right, yeah. I'm just going to mention that, actually.
Okay.
Yeah, that's a good point, an interesting point, that all the mitochondria in a cell are of maternal origin, okay? Now, okay, the chromosomal disorders are chromosomes then are distinct structures. You've seen, I showed you them, made of DNA and a variety of proteins. Some of these are histone proteins. They bind to DNA. There's other proteins. Histones are small basic proteins that bind to DNA. DNA is acidic. Histones are generally basic, okay? But there's a whole variety of non-histone proteins that also complex with DNA, and these play a role in the packaging of DNA into the chromosomes and also in the regulation of gene activity. And there are 23 pairs, as I said, of chromosomes. They carry most of our genetic material, apart from a little bit in the mitochondria. And abnormalities of chromosomes have been well recognized. Extra copies, breakages, abnormal rejoining of chromosomes in an abnormal manner, and these are called transpositions. Some can be detected by microscopic examination, and I think most of us are familiar with Down syndrome, in which there's an extra 21 chromosomes. So these are common disorders in which a lot of, for example, in Down syndrome, a lot of research is necessary to see exactly how the extra chromosome produces its effects on the individual. Okay, now to come back to Dr. Marvin's point, we have mitochondrial diseases. These are relatively rare, and as he pointed out, they are transmitted from the mother. All the mitochondria in a cell are transmitted from the mother. The ovum has the mitochondria, whereas the sperm is small and has relatively few mitochondria compared with the relatively large ovum. Okay, so these are due to mutations in mitochondrial DNA, MT mitochondrial, and amazingly the structure of human mitochondrial DNA has been known for 20 years or more, and it's known to contain 16,569 base pairs. The entire sequence of this was established for quite a while before the Human Genome Project, because it's a much smaller molecule, 16,000 compared with the 3.2 billion base pairs in the nuclear genome. And there are 5 to 10 copies of mitochondrial DNA in a mitochondrion. There's a variety of mitochondrial diseases. I'm not going to mention them all, but one is Leber hereditary optic neuropathy, which affects vision. And there's been interesting results obtained in gene therapy in this condition in the last year or two. That's brought some optimism to the possible effectiveness of gene therapy. By gene therapy, I mean introducing a gene, a normal gene, into humans that will replace the abnormal gene. But that's not that simple, because we want the normal gene to go exactly, ideally, to the site on a chromosome where the gene is, and to get rid of the abnormal gene and to insert the normal gene exactly into the correct site. If it's inserted into some other site, that can lead to problems. For example, a few people already who have children who have had genetic gene therapy have developed cancers. It's presumed to be due to the fact that the gene, the normal gene that's been added, has, although it's functioned, it's been shown to function, it's been inserted into part of the chromosome where it disturbs the function of other genes, and that can have unfortunate consequences. Now, the mutation rate of mitochondrial DNA is much higher than that of chromosomal DNA, because there are no repair mechanisms that I mentioned earlier, DNA repair, and also there's a very high concentration of reactive oxygen species inside the mitochondria, because the mitochondria is the place where oxygen plays a particular role in making energy. Okay, so that makes the mitochondrial DNA more susceptible to damage. Now, there's a lot of interest then in these multifactorial genetic disorders, and these are complex, much more complex than the single gene disorders and the mitochondrial disorders, because they're due to a combination of environmental factors and mutations in multiple genes. For example, breast cancer susceptibility genes are found in a number of different chromosomes, and these diseases are more difficult to analyze because there may be the contribution of even 20 or 30 genes together. Slight variations in these genes contribute to the causation of some of these chronic diseases. Okay, some of the most common chronic disorders are multifactorial heart disease, high blood pressure, Alzheimer's disease, arthritis, the diseases that we hear more about and which we're susceptible to when we age. So this is a very important area, is to try to examine the genetics of these particular conditions. Now, many heritable traits are in fact multifactorial: fingerprint patterns, eye color, skin color, etc. Okay. Now, an interesting question is, some people say, why do we have genes that cause diseases? Okay. For example, the genes responsible for the iron storage disease that I mentioned are often called the hemochromatosis genes, because they're involved in the causation of hemochromatosis. But in a sense, this is a misnomer. Okay. The function of the genes that are involved is in fact to help regulate the absorption of iron from the gut. Okay, that's abnormal in hemochromatosis, but their function is not to cause hemochromatosis. But if they are mutated, if they're changed, that causes abnormal forms of their proteins which no longer control the absorption of iron normally, and that leads to excessive absorption causing hemochromatosis. So we shouldn't really speak of hemochromatosis genes. We should say genes that are involved in the causation of hemochromatosis. It's rather a subtle point, but some people say, why do we have genes that cause diseases? Well, it's the abnormalities of these genes that cause a disease. The normal gene is doing a very important function in the body. Okay, just a few of the ethical, legal, and social implications of the Human Genome Project. Ethical, legal, social. Are patients being properly informed about the risks and limitations of genetic technologies? That's an important issue. If you have gene therapy or contemplating it, are you informed of all the possible risks? How does personal genetic information affect an individual and society's perception of that individual? If you have a mutation, an insurance company gets hold of that information, can that affect the premium that you pay, etc.? Okay. Then who owns and controls genetic information? Okay. The Human Genome Project wanted most of the information to be put into the public hands, but there certainly are genetic tests in which there are patents, and some companies are for example, to be tested for breast cancer susceptibility genes is relatively expensive. Now, the price of sequencing DNA, I didn't say much about that. The instruments are very formidable instruments that can sequence thousands and thousands of bases a day, okay? And the price of sequencing is coming down, so it's now quite possible that individuals who are listening someday may have their complete genome sequenced. Some individuals already have had their genome sequenced. For example, Dr. Watson of Watson and Crick has had his genome, and other people have had their genomes sequenced.
Greg Vitter had his. He owns a company that does that, Dr. Murray. And
yep.
He estimated his cost $70 million.
Really, yeah. Now they're talking about it not too long being $1,000.
Wow.
But I don't think it's reached that yet, but it's coming.
Yeah.
Who is the person you mentioned?
Greg Vitter.
Okay.
he wrote a book entitled A Life Decoded. And it's a very
yeah, he was one of the major. Is that Craig Venter?
yes.
Yeah, he was one of the major contributors to the progress of elucidating the human genome. He was in fact competing for a while with the government organization, and he invented or made a lot of the machines, sequenator machines, and he really was a tremendous spur to the project being completed earlier than it was anticipated.
He was a big competitor with Francis Collins, who was
That's right. Francis Collins was the leader of the government side. Venter, I think, initially wanted to have a lot of patented rights to it, whereas my own bias, I guess, is that genetic information should basically be public. But
absolutely.
That would be my. Now, one other point Dr. Marvin mentioned earlier, I'm not going to have time to talk about it tonight, but there's a lot of interest in the area of epigenetics, okay?
Right.
And he may mention that in his talk on Monday, but epigenetics means that the sequence of bases in DNA is not changed, okay, but you can alter the bases by adding, for example, a methyl group, CH3, onto certain sites of the bases, and this can silence genes, or if you take off the methyl group by enzymes, this will activate the genes, and this is becoming a very important area of genetics, epigenetics. These are additions onto DNA, but you're not fundamentally changing the sequence of the bases, but you're modifying the bases, which changes the activity of genes. It can turn off genes or turn on genes. So I'm not going to say much about that, but you will meet that term increasingly. I think, as Dr. Marvin pointed out, there was a whole issue. Was an issue. Was it Time Magazine you mentioned?
Yes, January the 18th, Time Magazine.
Time Magazine. Are you going to discuss that a little more in nutrigenomics?
Yes.
Yeah. Okay, so in summary then, DNA is the genetic material in humans. The great bulk of our DNA is in our chromosomes. A tiny amount is in our mitochondria, but that's important. The double helical model of DNA, Watson-Crick model, is a key contribution to genetics. The Human Genome Project has been a huge contribution to genetics and other related areas of medical science. It's had many spin-offs: medicine, biology, nutrigenomics, the pharmacology, etc. For many, the next major step forward will be understanding of the causes of all the genetic diseases, including the complex diseases that I've talked about, and the development of methods such as gene therapy for curing genetic diseases. Now, much of the information in this webinar is derived from this site here, Government Genomics Energy Gov, although the information is in many modern textbooks of genetics. The figures were taken from our textbook. The Wikipedia and Mayo Clinic website, for example, contain much relevant genetic information. If you enter genetic websites onto google.com, this will give many excellent sites. And if you're interested in purchasing a medical genetics textbook, probably the best thing is to go to a university or medical school bookstore or a medical school library and have a look at a textbook of genetics prior to buying one. You probably want to buy a relatively shortish one. Some of them are quite huge. So I thank you for your attention. I hope that you were able to get understand the major points that I was trying to convey. And certainly genetics is now a central area of biomedical science. The advances have been tremendous. When I started medical school a long time ago, the structure of DNA had not been elucidated, and now we see that the complete sequence of our genetic material has been elucidated. Who knows what the future will hold? Thank you.
Thank you, Dr. Murray. That was tremendous discussion. I just wanted to say, when I went to medical school, I benefited from the double helix, which was in 1953. I graduated in 1956, so I had 3 years of it, which at the time was kind of confusing to us because they popped a whole new way of thinking onto us about our sophomore year.
We graduated in the same year, it seems, then.
Really?
Yep.
Well, I guess that's why we're good friends. Let me see. Let me see if I've got any questions here. Oh. Well, I don't see any, David.
Maybe nobody's listening.
Oh, yeah.
There are quite a few. Quite a few. There's one that says, what causes, you probably answered this, but it says, what causes deletions, insertions, or duplications?
Yeah, that's a good question. I don't think they precisely know at a chromosomal level. Obviously, if there's a strand break, then there's a possibility of abnormal rejoining, but not nearly as much as known about these mechanisms, because they're not. I don't think there are good animal models for studying these. So I don't know too much, to be honest, about the mechanisms involved in duplications. There's probably geneticists out there who could do a much better job of answering that question. The most detailed information the most information that I have studied has been on the monogenic disorders such as cystic fibrosis, hemochromatosis, sickle cell anemia, etc. These are quite well understood, what the change in DNA is and how that affects the protein, etc. But the recombination of chromosomes is still an active area that I think a lot has to be, there's still a lot of discoveries to be made.
Okay.
I think that also the environment and your diet and your lifestyle, they've found, make some changes in your DNA. That's why identical twins, you can have one with asthma and the other one doesn't have asthma. And even one with cystic fibrosis and the other one doesn't. And they now feel that that's what I've found in the study of nutrigenomics, that a lot of those things pop up that way.
Yeah, I don't think the genome of identical twins is exactly identical. There are slight variations, although overall they're obviously more similar to each other than any other human genetic material.
You mentioned, go ahead.
Dr. Murray, how can they be, my question is, and this has puzzled me for a while as well. I mean, how can they not be identical if they split from the same egg, if they came out of the same fertilized ovum? How is it that they cannot be cannot be exactly the same? Is that because of the methyl groups?
Well, I think there's slight differences between, obviously there's differences between the maternal and paternal DNA, and in some cases the two strands of DNA might not be exactly, they're not identical but complementary. I may not be answering that question that well. Maybe Dr. Martin wants to chip in.
David,
Yes, sir.
one of the things, I have a friend who has identical twins that aren't identical. Now, if you look at them at a distance, they're identical.
Yeah.
But the closer you get to them, the differences are very obvious.
Mm-hmm.
And I'm quite sure that genetically they are different. And the thing that I wanted to mention, started to mention about epigenetics, that I found out that was real fascinating to me was the fact that you have a person that is a heavy smoker and not in regular genetic changes, but in the next generation can have a shortened lifestyle. is children. They've
Yeah.
tested this up in the north of the Arctic Circle in Sweden, and they've done quite an extensive study. 1 year, these people have a gluttonous amount of food, and they eat and eat and eat. And their offspring, they've studied, have shorter lifespans, up to 35 years less than the offspring produced after they've had a heavy famine and no food at all practically for 6 months at a time. Their offspring have longer lifespans. It's a preservation of the race, and it plays genetically. And that's all. Go ahead.
No, that's good. I was going to say that you mentioned, I think, epigenetic changes, and I think they could play an important role in the differences between identical twins. That if one is exposed to different environmental influences, that could influence the methylation of a gene and turn it off, or conversely turn it on. So I think epigenetics will contribute a lot to the understanding of these differences.
Right.
And it's almost immediate. It's the next generation. That's what really was fascinating to me. And I looked at all these things on gene expression like a whole set of switches, like in your circuit breaker. You know, you have some turned on, some turned off. Well, different things happen, like lightning hits close to your house. Well, it'll turn some of those off. And that's kind of the way the environment and the food affects us, and our nutrition affects us in nutrigenomics, because it turns some of the switches off. But this new research that come out of northern Sweden is fabulous to prove nutrition has a direct effect in one generation on the livelihood of their children. That's amazing to me. I have one thing here, other than a note that Dr. Marvin's breathing heavy in the background. And I want to thank Gary for that, because I'm in a cold room and I was trying to keep warm. Anyhow, somebody said only a master creator could possibly form all of this for life. Don't you agree, Dr. Murray?
I would say yes. I guess there's different scientists have different ways of looking at it. Certainly, Francis Collins, who was the leader of the government contribution to the Human Genome Project, he's now the director of NIH, and he's very much a Christian, etc. There's I don't want to get into the discussion of evolution, etc., but I certainly think that most people would think that there's certainly a very important element of direction in these phenomena.
Right. And by the way, if anybody's interested, Dr. Francis Collins' book is entitled The Language of God. And, have you read that, Dr. Murray?
Yeah, he is. He's been in debates with the other side of the people who are. I think, for example, Dr. Watson and Watson and Crick are fundamentally what I would call evolutionists. Okay.
Yes, I think so.
Yeah, whereas Francis Collins is now whether God. I don't want to get into this to any great extent, but whether some people, I guess, would argue that God could work through evolution. Obviously, if that's another aspect of the debate. I'm not sure that if that is Collins's view.
Dr. Murray?
Go ahead.
While we're talking, someone asked you to please show the summary page that you had, one of the slides.
Yeah, okay, can I go back to it?
Yeah, you might try that.
Yeah, there it is there. Is that it?
I think that's what she wants, yeah. Thank you.
Yeah. Okay, so I think that's the gist of what I was trying to get across. And Dr. Marvin is going to talk, for example, about nutrigenomics. I didn't say anything really about epigenetics. That is becoming an important area. But I want to stress that the fundamental sequence of DNA is not changed. Epigenetics means modification of the bases, and the major modification is methylation, that is adding a CH3 group onto different places in certain bases, and that will help to turn on, to turn off certain genes, to silence them. And if you remove the methyl group, an enzyme can remove the methyl group, that can help to turn them on again. So that's one important area of epigenetics, but that is becoming a big area. In cancer cells, I think differences of methylation have been known for quite a while, and how much that contributes to the behavior of cancer cells is under investigation. So, but fundamentally, the basic structure of the DNA, the linear sequence of the bases, has not been changed. It's just modification of the bases, but also histones play a role too in silencing or helping to turn on certain genes. So they're part of epigenetics. These are the basic proteins that are tightly complex to DNA.
I have another question here. It's concerning the glycoproteins that are on the surface of cells.
Yes.
Are the enzymes responsible for those and their configuration, or whether they're there for their transplantation?
Yeah, the glycoproteins, basically, the protein part is coded by a gene. An RNA is made, okay, then that RNA makes a protein, then the sugars have to be attached to it, and usually the sugars are added on in the endoplasmic reticulum and in the Golgi apparatus. The Golgi apparatus is a complicated network inside the cell where many of the enzymes that add sugars onto protein molecules to make glycoproteins are present, so that all the sugars that are in on a glycoprotein, some glycoproteins may have just one sugar, some have a long chain of maybe 10 or more sugars. One protein on the red cell has 16 different sugar chains, but almost all these sugars are added on by enzymes. There's a different phenomenon in diabetes. If your blood glucose is high, the glucose will react with hemoglobin, but that doesn't need an enzyme. It's a strict chemical reaction. If some of you out there are diabetics, you'll get your hemoglobin A1C measured, but that's not an enzyme reaction. It's actually a strict chemical reaction where glucose reacts with hemoglobin. Here, for glycoproteins, all the sugars in a chain are added on by a battery of enzymes that are present in the cell, and ultimately each enzyme has a separate gene.
Dr. Murray, I have a little fact that I found in the study of epigenetics with my nutrigenomics, and that's that TCF7L2 is a gene they've now identified as the direct epigenetic cause of type 2 diabetes, which I thought was fascinating. And they say if you have one copy of the TCF7L2, you'll 40% chance of being a type 2 diabetic. However, frequently we see two copies of that, and it's 140% chance of becoming a type 2. And I think that answers a lot of questions, because there's a lot of people that it seems like that most of that family is just prone to have type 2 diabetes.
Okay, but what percentage of adult of type 2 would that mutation or variation account for?
It's supposed to be very high.
Okay.
It came out of this Swedish research. It's supposed to be very high, maybe 60, 70%.
Really? Yeah.
Yeah. in October, I was at Harvard and attended their Genetics in Adult Medicine conference.
Yeah.
And they're going to try to have Dr. Watson speak there next year. But anyhow, I saw there, every field of medicine, whether it was ENT, GU, GYN, any field, GI, cardiovascular, pulmonary, all of them have involvement in genetic expression. All
That's right.
diseases.
Yeah.
And
Genetics has become a focal point of medicine. Ophthalmology, there's genetic diseases.
Yes.
Even healing, there's genetic diseases too.
Macular degeneration.
Macular degeneration, yeah. Even cataracts, there's genetic contributions too.
Sure. And you know, there's 6,000 genetic diseases now. And we call them diseases, but they're really not diseases. They're malexpression of the genetics in their body. You know, they don't really have a disease, except that's the only thing we can treat it like. But if they have a trigger point, and there's the epigenetic stimulated, then this can be reversed with nutrition, lifestyle, and, you know, how we conduct ourselves. I think that's the most fascinating thing in medicine.
Yeah, my own opinion would be if there's a mutation, it's going to be hard to change that mutation back to normal without some type of genetic treatment. You may be able to compensate in certain diseases by turning on another gene or something like that, but you know, in sickle cell anemia, the mutation is exactly known. One base is changed to another. That changes one amino acid in hemoglobin. That results in sickle cell. But there are ways of helping people with the disease. But ultimately, to my way of thinking, the ideal would be to change the abnormal base back to normal. And I'm not sure that a drug or a nutrient would do that. That's my opinion.
Well, here's one thing that's fascinating to me, and you know, I think you and I should do a presentation to a group of doctors, because I think it would really be a good service, because I don't think many doctors have gotten into it, and I wouldn't if I wasn't retired from regular medicine and was fascinated by it. But one of the things they're working on now is a virus that will go in and attack the cancer cells, put out a protein that protects them from macrophages and the body's defenses. It's their own protective self-insurance. And this virus will go in and kill that mechanism of their protection. And these tumors have just disappeared, like in 2 or 3 months, in the test cases that they've been testing them with. And it takes a different virus for different tumors.
I see. Yep.
They're making cultures of these, and the hopes of the future is to give a person with cancer, say a melanoma as you mentioned, a certain virus that will go in and destroy that cell's ability to preserve itself from the host's immune system. Therefore, as soon as that's stopped, that tumor will just disappear no matter how big it is.
That would be fantastic if it comes to...
yeah, they've got 1 or 2 viruses now that they're doing research on.
Yep. Yeah. The progress is amazing.
Isn't it? It's. I wish you and I were just getting out of medical school.
Yeah, we'd probably have big debts to pay.
Yeah. Nowadays, I had big enough as it was. Well, anyhow, it's been great. Of course, I'm prejudiced. I really love to hear Dr. Murray, and I love to hear this presentation. And I want to thank you for that resource page where people can look at Wikipedia and genetics from the government and all those websites.
The government site is excellent.
Yeah.
Yep.
I've got about 40 sites, genetic sites, that I've found. And
some of them are first rate.
Dr. Marvin, someone is asking, is there a source we can access for that northern Sweden study that you mentioned?
Yes, there is, but I'll probably have to give it Monday night. how you can access that source.
Okay.
Some of the words I couldn't even pronounce because they're Swedish, and I've been in Sweden, but this pretty near blew me away. And I'll have to dig those out, and I can, whoever it is that asked the question, I'll try to give that Monday night when I do my webinar on nutrigenomics.
Okay, great.
And I promise I won't breathe heavy. I get excited hearing Dr. Murray, and so I start breathing.
I hope you can make out the Scottish accent. I should have bagpipes playing.
No.
Well, if you're going to have bagpipes, then you got to wear skirts then. Oh, I'm sorry,
Yeah.
kilt.
Thank you very much, David and Norman, for your contributions. Thank you.
Thank
hope
you,
you'll
Dr.
be-
Thank you for listening.
I hope you'll listen on Monday night.
I shall endeavor to.
You can correct me.
I wouldn't do that.
Thank you.
Thank you very much, Dr. Murray and Dr. Marvin. This has been great. Really
Okay. Au revoir.
Au revoir.
All right, folks. Good night, and have a great weekend. Try to stay warm if you're in the Midwest, and we shall see you on Monday.